First of all, we are going to draw the diagram of the CS of a leaf. CS means cross section. This circled part of the leaf is magnified in this diagram, showing the upper epidermis of the leaf. Epidermis is formed of a single layer of cells. It has less stomata because if there is more stomata, more water is lost through transpiration. So only less stomata is shown in the upper epidermis. The work of upper epidermis is going to finish. Next, we are drawing the palisade cells of the mesophyll tissue. Mesophyll means middle layer or the layer between upper and lower epidermis. So, this mesophyll has two type of cells, the palisade tissue which is on the upper side and the spongy tissue on its lower side. Palisade cells are long as you see in the diagram. The next layer, the spongy tissue, as the name indicates, it looks spongy with a lot of air spaces because more stomata are found on the lower epidermis. So, carbon dioxide need to fill in the space between these cells. That's why the cells are loosely arranged, which gives a spongy appearance to it. So it is called spongy tissue. Last, we are drawing the lower epidermis. As I told earlier, the lower epidermis has more stomata. Only one stomata is shown in the upper epidermis, whereas two are shown in the lower epidermis. These are the bean-shaped guard cells that are located on either sides of stomata. Last, we are drawing the chloroplast of the cells. We know that the lower side of the leaf is pale or light green in color. So, only less chloroplast is shown in that part, means in the spongy tissue. Only dots are given. The upper side of a leaf is dark green compared to the lower side. It means that the palisade tissue just below the upper epidermis has more chloroplast compared to the lower spongy tissue. So, dark line is given to indicate chloroplast in it. Now we are drawing the diagram of open and closed stomata. So stomata has guard cells on the either side that are bean shaped or kidney shaped. Make sure that these two guard cells are almost symmetrical. These are the epidermal cells near the stomata and guard cells. They look like petals from the upper side. Also, they don't have any intercellular spaces. This is the nucleus of the guard cell. Each guard cell has one nucleus each. We are drawing chloroplast on either sides of the guard cell nucleus. This is the differential thickening of a guard cell. Guard cell is thicker on the inner side, whereas the outer convex side of the guard cell is thin. Next is the diagram of closed stomata. Guard cells of the closed stomata will shrink as they have less water. So, it is drawn very thin compared to the guard cells of open stomata. I am drawing the epidermal cells as flower petals. Same as in the last case, the guard cells are given nucleus and chloroplast. The differential thickening is given. Due to this differential thickening, the stomatal aperture is closed.